Hello students, in today's class we are going to learn about chemolithotrophy. Now earlier we have learned about electron transport chains wherein organisms make use of sugars to generate reducing equivalents, and then these reducing equivalents provide the required electrons for the terminal electron transport chain to generate energy. Uh, however, in case of chemolithotropic organism, these organisms, instead of making use of organic molecules like other organisms, they make use of chemicals as a source of electrons to generate energy in the form of ATP. Again, the chemolithotropes can be autotropic in nature or they can be heterotropic in nature depending on the conditions. So autotropic means they can fix their own carbon dioxide to make sugars. So here in chemolithotrophy, we are going to discuss about three examples, that is hydrogen oxidation, ammonia oxidation, and nitrite oxidation. So looking at the first example, that is of hydrogen oxidation. So enzyme hydrogenase is involved in the process. And in case of microorganisms, two different types of enzyme hydrogenases are present. The membrane-bound enzyme hydrogenase is involved in generating proton motif first and thus making ATP, while the soluble hydrogenase is the one that is involved in generating reducing equivalents. So here you can see that in case of membrane-bound hydrogenase, it will extract protons and electrons from hydrogen and transfer the electrons through a short electron transport chain involving quinones. From quinones, the electrons are transferred to the cytochrome B, while the protons are excluded across the membrane. From cytochrome B, they are transferred via C cytochrome to the cytochrome AA3 complex, which is involved in reduction of oxygen to water. And during this step, the cytochrome AA3 complex will also exclude additional protons across the membrane, contributing to generation of proton motive force, which is utilized by ATP synthetase to make ATP. Then we have the soluble hydrogenase, which will also extract protons and electrons from hydrogen and will use it for reduction of NAD plus to NADH. NADH is required in the Kelvin cycle to generate for CO2 fixation and to generate cellular material. However, if fixed form of sugar are already provided to these bacteria, then the activity of soluble hydrogenase is reduced, and only the membrane bound hydrogenase is more active, generating energy in the form of ATP. So here this next cycle describes the process of ammonia oxidation and how energy is extracted during ammonia oxidation. So here, instead of sugars, ammonia is oxidized, and as you can see, the enzyme ammonia monooxygenase, which is a membrane bound enzyme, is involved in oxidation of ammonia into hydroxyamine and water. Hydroxyamine and water will then diffuse from the cytosol into the periplasmic space, where they are further oxidized by hydroxyamine oxidoreductase. In the process, protons and electrons are extracted. Overall, 4 electrons are extracted, which are transferred to a C cytochrome. Now, from C cytochrome, 2 electrons will shuffle back through quinone to ammonia monooxygenase enzyme, which is involved in reducing ammonia into oxidizing ammonia into hydroxyamine with the help of the reaction that is represented here. So you can say that out of the total 4 electrons that are generated during oxidation of ammonia, 2 electrons are actually used up in the step, and only 2 electrons are transferred further through the cytochrome C again to cytochrome AA3 complex, which will convert oxygen to water in the process, contributing to generation of proton motive force, which is utilized by ATP synthetase to generate ATP. Now, the last pathway over here is of nitrite oxidation, and it is employed by nitrite oxidizing bacteria, which make use of the enzyme nitrite oxidoreductase to convert nitrite into nitrate. So in this process, electrons are extracted, which travel through a very short electron transport chain involving a cytochrome C and cytochrome AA3. So only at this stage there is exclusion of exclusion of protons, wherein cytochrome AA3 complex is involved in reduction of oxygen to water. So that's it about Chemolithotrophy, and thank you very much. Kindly like, share, and subscribe if you like the contents and to have direct access to the videos.